Hi, this is Miss Litton, and this is my wonderful Period 3 AP Bio class. Say hi.
Hi.
And they are motivated on this Friday, and we're going to talk about Investigation 13, Enzyme Activity. So the first thing here, it says, how do abiotic or biotic factors influence the rates of enzymatic reactions? Well, the first thing I would ask you is, without even doing this lab, what did I tell you? What did we discuss in the presentation? How enzyme activity can be affected? By what types of things? Yes. Heat. Temperature. Yes. pH. What else? Amount of substrate. What else? Amount of enzyme. What else? More. Cofactors. What else? Inhibitors. Good. I was inhibiting her. All of these things can affect enzyme activity. I am making that claim. Your textbook is making that claim. Your lab book is making that claim. But whenever you have a claim, you need to back it up with what? Evidence. This lab is an opportunity for you to get some evidence to support that claim. Okay? And in this lab, our reaction is taking hydrogen peroxide and using the enzyme catalase to break it down into water and oxygen gas. I think I have discussed this with you before when we talked about peroxisomes, yes? And how all living tissues have catalase in them. And so if you get a cut and you pour hydrogen peroxide on it, what will it do? Now, is it bubbling because there's bacteria there? No, it's bubbling because your tissue that you just cut has catalase in it. And if it stops bubbling, that means it's not making any more oxygen gas. Why do you think it stops bubbling? Probably you depleted your cells of catalase, right? You ran out of enzymes, so you didn't get that product anymore. Okay? Or another reason it could stop bubbling is you ran out of hydrogen peroxide. That would be another reason. So in our lab, we can do it different ways, but I'm going to demonstrate for you modifying how much substrate we have. And that's the part we're going to change, bless you. So in this lab, you are going to take hydrogen peroxide. And can I get eyes up here, not on your lab maps, but up here? Hydrogen peroxide, and you're going to add the enzyme catalase. It's not yellow. Every fluid today is clear. Every fluid, okay? This is why you have to be very, very OCD today. I hope you didn't take any medication. And I want you to be focused, and because if you mess up the syringes or the solution, your lab will be messed up for sure, okay? Because they're all clear solutions. You can take your hydrogen peroxide, you're going to add your catalase, and that's going to generate water and O2. And what you're going to use in your lab today is you're going to use probeware that can measure the amount of oxygen in your beaker, okay, in the parts per trillion, okay, parts per trillion. And you're going to get a rate of how much oxygen is generated per minute. Now, you will do this lab for 3 minutes. So you'll take your final minus your initial and divide it by how many minutes? 3. And then you'll have how many parts per trillion of oxygen you generate per minute. You'll get a rate. And we'll be able to compare the rates of those reactions. Okay, ask your bio buddy, do you understand that so far? Did you read your background? Do you get that?
I totally understand.
Okay, so I'm going to show you how to set up the collection of that data with the probeware in just a minute. I'm going to kind of keep going through this first. So your independent variable in this case are the concentrations of the reactant, hydrogen peroxide, but we could have changed the temperature of the enzyme, or we could change the concentration of the enzyme. These are all your things you could change. Your dependent variable, though, every time is going to be the amount of product, because we can measure how much oxygen is generated. We'll keep things the same. Your substrate, if we vary that here in this lab, we could have a 3% solution, we could have a 2%, we could have a 1%, a 1.5%, but either way, the initial volume is always going to be the same, and that's going to be 5 mils of substrate. The amount of enzyme we use, regardless of its temperature or concentration, will always be how much enzyme? 0.5. Now, some of you have started typing. This is available to you later. Right now, I want you to focus and listen into what I'm doing and not do the typing right now. You can always come back to this later, okay? So you can do, where am I doing? I have it somewhere. I said 1 mil here. We're going to do 0.5 mils. 0.5 mils, okay? And And not time 3.5 minutes. I'm going to change it. We're going to do it for just what? 3 minutes. Good. And your control, if we're going to do hydrogen peroxide when it comes, like I bought it at Vaughn's, okay, for the lab today. And it comes as a 3%. And just to move things along today, we're going to do a 3% and we're going to do a 1.5%. But our control will be not using hydrogen peroxide, but instead using what? Water in place of that. Does that make sense? Okay. So I'm going to model this for you to show you how you do this. But you have these containers right here, okay? And this is station 1's. It probably has something in here from the last class. So if they have any reactant left in there, it could throw off your results. So I would definitely rinse it several times, and then don't make a mess. Don't shake it over the floor, but shake it over the sink and try to get all the water out of it. Okay, so you have something that looks just like this, and then you're going to put your hydrogen peroxide inside of here. And if I'm going to do one, I personally would start with the control, so that way you won't have any residue in here. Okay, but just for the purposes of demonstrating this for you, in your boxes, you have 2 vials that look almost exactly the same. Okay, this one is marked H2O2. You see it? And the lid is also marked H2O2, so you don't mess the lid up with the container and possibly contaminate, right? And this one is marked water. And you have 2 syringes sitting like this because you don't want to contaminate them. I don't like what Team 1 did, because do you see how I put the syringes in here up? What did they do? Put it down. And if it happened to have the hydrogen peroxide in there, do you see how it could be contaminated? So I would rinse off the end of your syringes and I would store them like this. Okay? So I'm going to grab the hydrogen peroxide one out of here, and I'm going to pick up, I would carefully pick up 5 mils. Do you think it makes a difference if I accidentally pick up 6 mils? Why?
Per minute, so because that's what.
Think about it. And I'm trying to make a controlled experiment, right? So I want to know parts per million. Will it change the amount of product I have if I put 6 mils instead of 5 mils in here? Yes. Okay. So I'm just going to put in here, make sure I put in 5 mils. I'm going to be real exact with my syringe. And I have 5 mils of hydrogen peroxide in there. Now, before I put in my enzyme, okay, and by the way, your enzyme is labeled here. It says catalase on here, and you have a syringe that stays in here. Don't put it on the counter so it won't contaminate anything, won't pick anything up. Before I put that in here, I need to set all of my probeware up, and I'm going to show you how to do that, okay? Basically, this oxygen sensor right here gets connected to this air link that you see here, and you turn on the air link, and you Bluetooth it to the iPad. And so you're going to go into the iPad and to the settings, and you want to receive the air link that you're using, not the air link that somebody else is using in the room. All the air links have numbers on the back. Okay? Now, before I switch over to that, I want to just say I made a lab map for you. It was definitely a rough draft of a lab map, and you have a copy of that right in the lab map that I gave you. Okay? So I put a flow chart. It definitely doesn't have enough room in here how I did it, but I would recommend that you make a rough draft so when you put it in there, it's spaced neatly. This right here is one of your equations that you need to know. Besides rate of reaction, you have to be able to make dilutions. So what you do, you can, sometimes they write it C1V1 equals C2V2, or I for initial and F for final. So you can learn how to make dilutions. I usually use this lab, and we do a 3%, a 2%, a 1%. But for the purposes of moving this along, we're going to do a 1.5% as one of our variables. We're changing the concentration of the substrate. It comes as a 3%. So if I'm telling you to put 5 mils in there, what could you do to make it a 1.5% instead of a 3%? 2.5 of hydrogen peroxide and 2.5 water. Okay? I want you, using this equation, to double check and show me that dilution, how you would do that. I'm going to give you an example in here of a 2% solution. If I had wanted to make a 2% instead of a 1%. What you do is you take your stock solution and that concentration. Well, this stock solution of this hydrogen peroxide is a 3% solution, so I put 0.03. This is what I'm solving for. How much of that stock do I need to make my dilution, right? I can't make it more concentrated. I can't take a 3% and make it a 5%, right? Because I can't make it more hydrogen peroxide, right? It comes as it comes. I can only dilute it. And the dilution I want, in this case, was a 2%. And I want 5 mils of a 2% solution. So then I'm just solving for x. So 0.02 times 5, right, divided by 0.03, x equals 3.3. That means I need 3.3 of the stock solution, and then I bring it up to 5 mils, which would be 1.7 mils of water, would then give me a 2% solution. Now you are just using your logic to go, half of 3% is 1.5, so half hydrogen peroxide, half water. I want you to show me the math in your flow chart of how you would get that. And that way you can practice using, this is one of your equations you need to know for AP, all right? And then you're going to grab, you have a data table where you're going to put in 0% concentration of hydrogen peroxide. What would this be, our 0%? That would be our what? Control. What can we do to get 0% hydrogen peroxide?
Just use water.
Just use water. Good. Okay. 1.5%, that's when you do 2.5 water, 2.5 hydrogen peroxide, and then 3% is just straight what? Hydrogen peroxide. Okay? And then what you're going to do is then you'll have it at 0 minutes, 0.5, all the way up to 3 minutes. You will have your group rate, so you will have to do the math underneath here, parts per trillion per minute. So you'll take your final reading minus your initial reading, divide it by 3 minutes, and that's your rate. Okay? And you'll write your group rate, and then we'll get the course, what were the average rates. And you don't want to be outlier data, right? Okay, now, if we're going to do a null hypothesis for this, how could we write a null hypothesis for this? The concentration of the hydrogen peroxide does not affect the amount of oxygen produced. Okay? So let's say I'm doing my 3% solution, right? And I'm doing my 0%, which, right, is just water. Let's say when I do the water, let's say I get the rate of oxygen production. Let's say I get 1.7 parts per trillion per minute. Okay? That would then become my expected value in my null hypothesis, right? That's my expected value. And let's say I get 9 parts per trillion in my 3% solution. Now I take my observed, 9 parts per trillion, minus my expected, 1 point whatever I said, square it over 1 point whatever I said. That sounds like a big number to me. And then I could do that again. Another whole experiment is doing it at 1.5%. And you could see, is it statistically significant if we vary the substrate, the concentration of the substrate, does it vary the amount of product we have? That will give us evidence to support the claim that we can affect enzyme activity. Youngest bio buddy, yow in your own words, please. What are we doing in this lab? Go ahead.
Okay, so hydrogen peroxide. How much does that affect?
Can I really quickly run to the bathroom?
Sure. Really quickly, because I'm showing this right now. Okay. Now, I am airplane and my iPad now up on here because this is what you're going to be using, so you'll know how to set up your iPad to read these readings. Okay? So the app that you're going to look for is SparkView. It's like this one. SparkView, you see it? Yeah. But I am going to be needing to turn on the blue, before I even go into SparkView, I need to connect to the sensor via Bluetooth. So intuitively, where do you think I need to go in order to do that? Into settings, okay? Now, here is the oxygen sensor. Here is the air link right here. So you need to look on it because you want to not mess it up, and you want to put it in there the way you're supposed to put it in there. Okay? And then to turn it on, you turn it on here on the side. You turn it like towards the oxygen sensor. That's how it turns on. It should be green. Are you seeing any greenness? See it?
Yeah.
That means you're ready to Bluetooth it. Now on the back, there's a number. What number is on that Bluetooth?
15201.
15201. So I'm going into settings right now. Can you see it on the board?
Yeah.
Where do you think I need to go next? So I'm going on to Bluetooth. What do you think I need to do? Turn it on. Do you see it? 15201, so I'm selecting it. I'm now connected to this oxygen sensor. Okay? So now I'm going to go back and I'm going to find that app. What was the name of that app? I'm clicking on it. Okay, and I'm going to wait for it to figure, oh, there it is. Can you see the oxygen concentration on the board? Okay. Did it change a little bit?
But you actually CO2.
You're right. So it should go down. Did it?
Yeah, there we go.
Yeah, so it went down. Now, it's set at parts per million. Can you see how that's really a peta to graph?
Yeah.
So that's why I'm going to have you change it to parts per trillion, okay? So now, what I'm going to do is I'm going to set up a way to read it. So the way you want to do this is I make sure, I double check, it's reading my sensor, I'm good. Now I'm going to build. Oh, I always forget. That doesn't touch. Okay. Now, the way I would do is I would break it up into 2 equal halves over here. So I'm going to bring it up into 2 equal halves. And one, I want to have a graph, so I'm going to pick the graph button right there.
That's cool.
Okay? And I'm going to set up my graph. I'm going to select my measurement. So I'm going to hit that orange box that's lit up, select measurement. And my measurement I want to know is what?
Parts per trillion.
Parts per trillion. So do you see where it says O2 ppm? I'm going to click on that. Now do you see parts per trillion in there? PPT, parts per trillion. So that's my oxygen concentration here on my y-axis. What do I want my x-axis to be?
Time.
Time. Okay, so I'm going to go into time, and I'm going to check minutes. Do you see where minutes is?
Yeah.
Okay. Now the reason why I'm going to do it at minutes is because then I can take data down at 0 minutes, 0.5 minutes, 1 minute, 1.5 minutes. Okay? On the other side, I want to have a really nice data table that they make for me that I can use to look at and just write down in my own data table. So you see something up there intuitively that looks like data table?
The thing that's like under the box.
We're going to say here? That's what I agree. Okay? So now I'm going to select my measurement. I'm going to pick time, which is going to be in what? Minutes. Okay, and what's my other measurement going to be?
Parts per trillion.
Parts per trillion. You got it. Oh, cockapoo. Parts per trillion. Oh, I'm so glad I recorded that. Okay, so did I set everything up? Yeah, I feel good about it. Okay, now the next thing I need to do is to start the readings. The green button right here is like, what do you think that means intuitively? Play. Okay, so what did I put in here just a little bit ago?
Hydrogen peroxide.
Hydrogen peroxide. So now I'm going to add in how much catalase?
0.5 minutes.
0.5. This is a little 1 ml syringe. So now I'm going to put in 0.5. And then I need to be ready to hit play right away. Okay, so I'm putting it in. I'm going to put on this. And I'm hitting play. And I am taking data.
You don't even have to do anything.
Exactly. How long do I run this data?
30 minutes.
Exactly. Now, I'm not going to run it for all 3 minutes right here because I want to show you something else. I'm going to take 2. I'm taking yours right now. And I'm going to put in not as much enzyme. or not as much substrate while that's running. H2O2, I'm double checking. 2.5. I didn't rinse this out, so that's risky. I'm just doing it for a demo. I'm gonna take some water. 2.5. And a 1/2 ml of enzyme. I've got it ready to go. Okay, now I'm ready to stop. So I'm stopping it. Did I go 3 minutes?
No.
No. How many did I go?
1.2.
1.28. Now, watch this. I'm just going to take the sensor off of here. And that's one, which I need to fix. Okay. Now, I just did half water, half hydrogen peroxide in this one. Got my 0.5 mils. Putting in my catalase. Don't put it on too tight. Notice it's starting a brand new run.
What?
Right on top of it.
Why did it start so much higher up?
That was its initial amount of oxygen that was in there.
But what? Uh-oh, that's not good.
Now, remember, this is why you do final minus what?
Initial.
I mean, I'm starting out here at 186 parts per trillion, and now this is 196 parts per trillion. Okay, so we're talking about very small difference, right? Okay. But now it's starting to go up.
The graph makes it a big deal. Yeah.
Now, you can say, well, I don't want, oh, I forget, it doesn't want me. I have to touch in here. I can go, okay, I'm going to take run 1 out and just look at run 2. So I'm saying, or I can click it back on now. I'm looking at both runs at the same time. Now, I'm going to just stop it. Okay? And now, here's the beauty of this. When I go to record my data, let's say I go back, oh, my bad. Go away. I'm going to go back and I want to see run 1. I click run 1. What did it start out with at 0 time?
196.
So I'm just scrolling back here. It started at 0 time, 186, right? And then what was it at a 1/2 minute? You see? Oh, I'm not there yet. Thank you. So I'm scrolling to a minute. Okay, it's really, it should just be doing 0.3. I'm kind of annoyed. I don't know why it's doing that. Oh, whoops.
This is a time.
I saw your hands.
Do you see it? 0.5, right? What is it doing it at 0.5?
189.
Yeah, 189. What did we start out at?
186.
186. So it went up 3 parts per trillion in how much? Half minute. And then I could go and see what is it at a minute, right? And I could write that one down, which I just passed.
That's okay.
I know, you're like, Miss Litton, don't take the time. Okay, so what are we at a minute?
That's 592, right?
Like 192. Okay, you can switch and find out your same data at run 2, and you can see what and calculate the rate from there as well. Okay, so you're doing water, half catalase, and then what?
Hydrogen peroxide.
I mean, half water, half hydrogen peroxide, and then all full hydrogen peroxide. Every single time, though, you're doing 0.5. And remember, you just hit the play button. You can run your 3rd run right next to, and you can have all 3 on there. It'll graph all of them for you. Okay? And then you hit the red button to hit stop. Got it? Okay, have at it.
Alright, we're gonna put that up.
We can use
One of you.
Keep 1 and 2. Rinse out your beakers because I just used them. Yeah, take it.
I don't like this.
Yeah. Okay, that's fine. If you want to grab somebody else.
It's a good thing you weren't here then.
Oh, whoops. Hope that was helpful.